All right, thanks for coming, guys. I'm Ian. I'm from Yale University. I'm in the Erika Edwards lab. I usually talk about trees, so this is my first time in physiology, so let's see how it goes. If you weren't at Carolina Hayduke's awesome primer on CAM today, I just want to refresh what CAM is really quickly. So CAM is reverse stomata behavior, which greatly increases the water use efficiency in plants. So what these plants are doing is they're taking carbon dioxide up at night, converting it, concentrating it into C4 acids, storing these acids overnight in the vacuole, exporting them during the day, and then decarboxylating them, which then allows carbon dioxide to enter the Calvin cycle as normal. And this is an extremely complex character, in that the transition from C3 to CAM involves a whole suite of changes, ranging from transitions in ecology to gene duplication and differential recruitment of paralogs. And all of these changes are happening under this crazy constraint that photosynthesis has to remain functional in the evolutionary trajectory of these plants. Which to me is why it's so fascinating that CAM has evolved dozens and dozens of times, and not just in concentrated clades, but really broadly across all of land plants. So how has this managed to happen? So today I'm going to talk a little bit about different CAM phenotypes, why we need to know them and why they're difficult to know when we study evolution of CAM. A couple resources that I've been developing to do some of this work, and then use those resources to show you a couple interesting observations using machine learning to disentangle CAM phenotypes. So CAM, like any character, to study the evolution, we want to put it on some sort of tree. But to do this, we first need to, one, agree on the different character states that we're talking about, and then be able to reliably delimit them. So we need to be able to actually go and measure them and look at them and put them on our tree. This is not so easy. Again, Carolina covered this a little bit today, but if we think very rigorously about the scale of caminess from less CAM, over here, taxa that are C3, exclusively fixing carbon during the day. On the other side, we have plants we know and love, like the cacti. These are things that are only getting their carbon dioxide and fixing it at night. In between, we have a couple intermediates. One, facultative CAM. These are plants that exhibit CAM physiology when they're droughted often, but just stressed in general. And also things that we call low-level CAM. These are things that are always fixing some C3 or some carbon at night and some carbon during the day. And you might think that the evolution from C3 to CAM simply proceeds on a linear fashion through these phenotypes, but some work that Willie Hancock, a previous member of the lab, has done has shown that we actually see a bunch of transitions happening among these intermediate phenotypes, as well as back and forth between these and C3. And we aren't so sure what actually brings these intermediate types all the way up to the realization of full CAM. The sort of gold standard in determining CAM phenotypes right now is carbon isotope ratios. So if we look at these here, we have C3 in yellow, CAM in blue. This works out pretty well. But as soon as we start looking at our intermediates, which I've just collapsed into this phenotype I'll call C3 CAM, they run the gamut here from C3 all the way up to full CAM. And the only way that we can actually get at these phenotypes is through a series of experiments looking at titratable acidity or looking at things like expression levels with RNA-seq. So one thing we started to think about was, can we look and find some of these characteristic morphological features of CAM taxa and place them along these intermediate states and see maybe is morphology correlated with these intermediate transitions in CAM phenotypes. So the question for the remainder of the talk is, do these phenotypes actually correspond to this morphology? And this, looking at this has gotten me to develop a couple databases that I've been compiling, and finally led to a small Python code that I've been using to understand this data and do some basic wrangling of it. All of it is available on GitHub. It should be pretty easy to download and install, let me know if it's not. And there's a Jupyter notebook that should really easily walk you through every single analysis that I'll show you today and some other ones. The two databases that are present in this package or just on the repository itself are called CAMDB in the list. This is just an artifact of the original database, which is just CAM taxa. It's been expanded for all types of photosynthesis now. And what we have here are a bunch of observations of about some set of up to 30 different morphological or physiological measurements, associated taxonomic information, and also some metadata on the publications. And in the list, we have a list of all plant genera pulled from the website Plant List. It's about 21,000 genera. And then for a subset of those, we have all of the observations of different modes of photosynthesis that authors have recorded in publications. The package itself lets you take this data, and if you keep it relatively in the same shape, you can get some really usable features out of it while you're working with it. So here we can visualize a plot of the missing data, and we can see that in this instance we have about 5,500 observations, 3,500 species, spanning about 60 families, 60 publications, and our matrix is pretty sparse, about 80% missing data, and only a few of our observations, 760, actually have a labeled photosynthetic pathway, which is often problematic for the work I'm trying to do. So for example, here we have carbon isotope ratios again, and we have our unlabeled individuals or unlabeled observations here in gray, and then labeled here with an outline, which are then color-coded on the right. With a couple of imputing functions, we can cross-reference these databases and look among species and among genera, and flip this so now we have many more labeled observations now eclipsing those unlabeled observations, and we cover this plot that I showed you earlier. So now with these data in hand and some tools for kind of wrangling it and turning the data around, we can start doing some things like machine learning, where we often need hundreds or thousands of data points to say anything meaningful. And again, the question is, do these phenotypes actually correspond to the morphology that we're observing? The first technique that I'm going to talk about is called support vector machines. And what we're doing here is we're taking our data with their associated labels or classes. We take those, we project them into a high-dimensional space, and we look for a plane that separates our classes with the most wiggle room in between. The next method is random forests, and it's really different. What we're doing in this case is we take a feature or a variable one at a time, and then we take all of our observations and we try and use that feature to split up all of our observations into those respective labels or classes. We do this iteratively through different features until all of our observations are sorted. We do this many, many times and create many, many decision trees. The first example that I'm going to show you is in the bromeliads. This is data largely from Jamie Males, and you can pull it really easily just by specifying the publication, for example. And our data here is about 160 species, 3 subfamilies, roughly divided equally between CAM and C3. We have a few C3 intermediates here, but we had to drop them just because there weren't enough to do any learning with. The features that we're looking at in this instance are air channel space, chlorancoma thickness and cell diameter, as well as hydrancoma thickness and leaf thickness. And part of the reason why we're concentrating on these sort of features is that these are things we associated with CAM being more succulent, having more space to hold C4 acids in your vacuoles overnight, and things like large cells with tight packing that kind of decrease the amount of carbon dioxide that could leak back out of stomata. A common thing we do with our data, almost everyone with any kind of data we generate, is we throw it into a PCA. And if we look at our PC space here, we see we've captured about over 80% of the variance, but we can say nothing meaningful about our different subfamilies or our labels here. And I can talk about why we see this later if you want. But with just these 5 features, we actually have remarkable accuracy in classifying things that are C3 and things that are CAM, with just these few morphological features alone. And with random forests, we have about almost 90% accuracy, so both these methods are performing very well, they're very robust. Here in these confusion matrices, we see that when we actually see a CAM taxon, we're actually predicting CAM, and vice versa for, or C3, vice versa for CAM. And there's only 3 measurements, or 3 observations where we're misclassifying them. The next data set that I took a look at was from Cactus silvera in orchids. Roughly the same size, but now a more even distribution of our classes. And here the features we're looking at are fresh mass to dry mass ratio, leaf thickness, and specific leaf area. So again, things that we can get pretty easily. As you might guess, we're not performing as well here when we add a third class and spread our data more thinly. And where this is coming from is largely C3 and C3cam are often confused. Our accuracy drops down to the mid to high 60s using our two methods. And if we look at our confusion matrices here, for example, with random forests, we're oftentimes encountering a C3 observation and calling, or a C3cam observation and calling it C3. And interestingly enough, we're calling lots of things that are C3, C3cam. So I tried probing this a little bit further. I dropped all of these intermediates, and we gained about 20 percentage points in either case. Then, strangely, we removed things that are strictly C3 and compared the intermediates to full cam observations. And while it's not significantly more accurate, we do increase in accuracy a little bit further, which is really strange to me. In sum here, what it looks like is things that are cam, really easy to distinguish from other phenotypes, regardless of what data set we're using and features we're using. C3 and C3cam are pretty hard to distinguish, though. They're almost undistinguishable in either case, although we can't really say much about anything in the Vermelia data set. So to conclude here, I hope that I've shown you that PhotoDB, this package, is useful. I hope that you'll use it or at least help me build it bigger. And what I find most exciting here is that morphology can reliably predict cam phenotypes. Again, these are the most extreme phenotypes which we know carbon isotope ratios work with, but now we have physiological measurements and morphological measurements both telling us the same thing with high accuracy, which is great to know. I was pretty bummed out about this result with these intermediates. I was not happy. I was like, I'm going to figure this out. I'm going to have this. I'm going to have these data. It's going to work. So could it be truly distinct with more data? I'm not sure. Is it truly indistinguishable from C3cam? We can't say. But it fits and doesn't fit with the two hypotheses we're sort of kicking around in the lab right now. One of them is that morphology may not be an important initial step in cam evolution. We may not need special morphology to do a little bit of cam when you're stressed out. You may not need these thick cells. Right. We may not need specialized anatomy. Although there was a really good talk today that seemed to say the opposite, also from a member of the lab, Carolina. So I'm really, really excited to grow this database more and hopefully get at this a little bit further. So again, adding more taxa to this is what I'm really excited, more observations, especially where we're having trouble confusing these. Adding also perhaps more data types like environmental or key genomic things that we see. There are certain mutations that seem to always pop up in these lineages when they shift to cam. But also some functionality. I'm, again, a tree-based person, so adding imputation that's based on distances that we see on phylogenies. And also some updated taxonomy, which is really important for these things like orchids or bromeliads that change every day. And with that, I'd just like to thank everyone in the Edwards lab and the CAM photosynthesis conference, which has really spurred a lot of this work.